and I'd like to welcome everybody back to the room after lunch. And I hope you all had a good lunch and particularly enjoyed the dessert table, which was quite impressive. So before we begin the session, first of all, I'd like to start by acknowledging our sponsors, who are a really big help and make all this possible. And that includes BASF and Bayer, our top sponsors. A big thanks to Nutrien, Manitoba Canola Growers, Sask Canola, Corteva, Alberta Canola, and also Canadian Canola Growers Association, New Seed, Syngenta, Western Grains Research Foundation, Winfield United, John Deere, and Decisive Farming by Talis Agriculture. So thank you very much to our sponsors. Just a reminder that we have the app, which is the way that you can send in your questions to the speakers at the end of the session. Use the question and answer function on the app, or if you're in the room, just stand up and wave and we'll bring you a microphone. Okay, so the first session this afternoon, we're going to be talking about the topic of abiotic stress. And as all we can remember from the 2021 crop year, drought was a particularly bad stress that particular year. It's still also an issue this year, maybe not quite so much in Manitoba where they were seeding with a submarine, but other than that, it's a stress that we have to consider in the long term for canola. So we've put together a session with 3 speakers who will address different aspects of stress tolerance and stress adaptation in canola, and maybe give us some clues as to how we can change and improve this crop. So without further ado, I'd like to welcome our first speaker. So our first speaker is, I always have trouble with your name, is Raju, hang on, Soolanayakanahally, sorry, good. So Raju works with me at Agri-Food Canada in Saskatoon, and I still struggle with his name, but yeah, he's a research scientist at Ag Canada in Saskatoon. His expertise is in woody biomass crops like willow and poplar, and also sea buckthorn. He has a very good collection of very tasty sea buckthorn, but he's also a plant physiologist, and they're becoming an increasingly rare breed. And he has a strong interest in abiotic stress, and he has been doing some very interesting work on canola in relation to drought and heat stress. And I will invite Raju up to the stage so he can begin to tell us about drought stress and what he's been doing with canola. So thank you.
Good afternoon, everyone, and I thank the organizers for this opportunity. This is first time I'm speaking in front of the canola crowd. So today I will be talking about abiotic stress tolerance with Brassica napus. Here I put together a team that's part of my lab, Rahid El-Farjani, who was a postdoc, Shankar Pahari and Branimir, who was research assistants in my lab, and Krista Thompson, who is our biochemistry technical assistant. First of all, I would like to make the land acknowledgement, mentioning that the Saskatoon Research Center is located on the Treaty 6 territory and the homeland of Métis. Today, we are collectively actively participating in reconciliation as we navigate our time in learning and supporting each other at this Canola 2022 meeting. Based on the input I received from the organizers, I was requested to give some kind of background on the climate culprits and kind of share what really went on in 2021 as far as physiology is concerned. And the second component I will talk is about some of the ongoing work in my lab related to heat and drought stress. So when we look at the CO2 concentrations, since the beginning of the Green Revolution, there is a steady increase in the CO2 concentrations going from anywhere between like 300, today we are at 420 parts per million. I gave 3 milestones. The year I was born, it was 330 parts per million. Then I moved to Canada, it was 380 parts per million. And today I stand in front of you all and it's 420 parts per million. Within my own lifetime of 45 years, we are seeing a big jump of like almost 90 parts per million. On the day one, when nitrogen activity was discussed, it was mentioned how important the nitrogen is. And there is more emphasis in government of Canada on reducing nitrogen emissions and other things. What I would like to bring today for this forum is like how CO2 concentrations inhibit nitrate assimilations. This is not at all discussed in the research community. The reason here is like under high CO2 fertilizer conditions, the nitrate fertilizers, the enzymes that are involved, both nitrate reductase and nitrite reductase, their enzymatic activity decreases. That means when the leaf is surrounded by high CO2 concentrations, these enzymes don't operate to their full potential. We can address this quickly and remove the bottleneck by transitioning to ammonia-based fertilizer. But ammonia is toxic to plant. Most of the ammonia has to be assimilated in the root system for bulk of the C3 plants and C4 plants. So we have to deal with toxicity. We can fix it quickly, but there is a catch here. If you use nitrate fertilizer, the plants produce natural defenses against pathogen. So far, if we have got resistance, there is a big component of nitrate fertilizer playing a role in that one. The moment we shift to ammonia-based fertilizer, the first formed product is GABA, and insect pests love GABA. So we can solve nitrogen problem, but we will get into disease problem. So we are in this catch-22 situation. In my program, when I first came like 5 years ago into canola research, the first thing I asked is like, in which direction breeders have been breeding for traits? Are they breeding for high nitrogen use efficiency all these years, or are they breeding for low nitrogen use efficiency? In my mind, there was other question. If I look at historical range of canolas, can I understand whether the plants have been selected for high CO2 environments already in the breeding program? So for this, what I did was, I took the plants from 1942 to 2017, a panel of open pollinated canola and hybrid canola that comes from all the commercial breeding companies. Then I subjected these set of germplasm to pure nitrate fertilizer and pure ammonia fertilizer. And I asked like, okay, how are you going to perform? Let me see. And I kept my nitrogen concentration very steady and said, I will give you as much as nitrogen. I keep the environment at its best. There is no management issue, no environment issue. All I wanted to know is the genetics, the G component. So it was very steady state. And I asked this question. What we found was most of the breeding programs are moving in response to CO2 conditions. And what they're looking for is enhanced ammonia utilization, not nitrate fertilizers. So this was something that came out. If this trend continues, we should be now selecting, if we want like better use of nitrogen use efficiency, then we should select or breed plants that will tolerate more ammonia toxicity in the root zone and also utilize ammonia better. Moving on, I deal with various kinds of stress here, from going from cold all the way to salinity. Stress is always complex. One, whether it occurs at vegetative phase, whether it occurs at reproductive, or it occurs at pod filling phase. And also the duration and intensity also matters. More recently, the studies that are coming and what we are seeing is combination of droughts where heat and drought come together, or when we go to southwest of Saskatchewan, heat, drought, salinity all come together. So it's getting much more complex than looking at individual stressors. Smoke is the latest thing that is gaining a lot of importance, but it is not that well studied within the cropping system. This was the beautiful piece of article that Javither put together on how the smoky conditions stalled canola growth in central Alberta. This was the photograph taken in 2018 during pod filling phase. And when we look at 2018 numbers here, that year we had more than 7,000 forest fires, and that engulfed the smoke, and that smoke caused tomatal closures, and most of the canola sat for 10 days without transitioning from vegetative to reproductive phase. Alberta farmers were worried, what is going on here? My crop is not moving. The very reason for that is like that year, we had the highest number of smoky days, 3 smoky hours, 230 smoke hours, which translate roughly to 10 days, and that was the delay we were seeing. We need to understand more. Already we are seeing the influences of smoke on California grape quality, where the smoke flavors are reducing their grape potentials and also growth potentials. In addition, we are seeing more extreme events happening. This is the carbon briefing article that composed more than 170 work, and they said events like heat, drought, flooding, cold might become more frequent as we transition to 2050 and beyond. Here I put together historical perspective of the droughts on prairies, going all the way from 1960, beginning of Green Revolution, to the most recent one. When we look at all these, there is a nice 20-year interval where these droughts keep coming. When we look 60s, 80s, 2000, and 2020. And when we look, the pattern looks very similar. They kind of mimic each other. In 2021, that was the worst drought in the last 60 years. Because of better agronomic practices, better genetics, we had just 40% crop losses. Otherwise, it would have been much more devastation. Looking at crop stress index here, this is a key indicator of how crop yields perform. And when we look at most of the canola growing region, either they came under severe stress or extremely severe stress. Severe stress was in the range of like 22 million acres, and extreme stress was in the range of 55 million acres that we experienced in 2021. I started to get a lot of like photos from Clint, Keith, Jay, and others like asking like, hey Raju, what is going on here? And this is one of the pictures that Clint and Keith shared from mid-August. This particular farmer was noticing like there is a new crop flush in the bottom parts from the lateral buds. What happened was, due to extreme stress, the shoot tips got pinched, and suddenly there was a rain in August, and the crop felt like it should produce new leaves, and the only mechanism they used was the lateral buds that were dormant. Why was it doing? Ecologically, all the canola plant was trying to do is it was desperate to produce its progeny for next generation. That was what was going on. Then this picture was the one that Jay sent, where he was observing some of the seed germination from the precaceous seed germination that was happening. And this happened due to the rapid in-pod drying that happened, and the seed embryo couldn't build enough ABA to inhibit germination. So when the rapid dry down happened, ABA accumulation was not adequate enough. Since they both coincided, that led to the precaceous germination. This is one of the classical examples of how evolutionary mismatch happens at a physiological level. So that all led to yield reductions. Here I have compiled like the last 5 years of yield with 2021 yield, and we can see a big drop in yields. And the oil fell, whereas protein went up. I really like to see one day the harvest index of canola improves. Right now, it really sucks when I compare all the C3 crops. It's not the best for the inputs we are putting in. Moving on, when we look at the climate projections for prairies, here I'm just showing like the projections for Saskatoon up to 2050 and beyond. We are anywhere going to get like 18 to 20 frost-free days. And our growing season temperatures, we are going to see more 30 plus days, which will be more hotter and drier. And crop water needs will be much more. So the research in drought is not very new in prairies. Heat down is started this long back. This was the report I pulled from Sask Canola reports. And Keith Downey and Natella did this report in 1992, but the work happened from 89 to 91. And what Keith did was that time, they went all the way up to 26-degree temperatures, and they used different precipitation ranges from 80 to 400 millimeters. And they were able to show how the temperature and reduction in water leads to reduced yields in Western canola. Keith, I was still in high school when you did this work. And then I went and said, was Keith really good at his math when he did all this work? Then I took 20 models, and the models exactly mimic what Keith has shown in 1989. Here I have drought severity index. The baseline used is 2016 to 15 with a 0.5 degree differences. And when we look at Saskatoon alone, it goes from like the stress index of 49.3 to 52.8, whereas yield losses go from anywhere between 6.6 to 8.2. And that is due to, this model took into consideration the CO2 levels as well, the CO2 concentrations. But if we don't take CO2 concentrations into picture, this would be much more devastating. Moving on to my own work. This is the work funded by Sask Canola and Agriculture Development Fund. Here I try to utilize some of the NAM founder lines that Isabel Parkin, Steve Robinson, and Sally Vale have put together. And this is one of the best diversity set to work with when we are looking for resiliency for canola. Here, I wanted to mimic some of the natural conditions. So I used 65 kilos of topsoil and grew 6 plants per variety and subjected them to well-watered conditions at 23 degrees, droughted at 23, heat at 29, and heat plus drought at 29 degrees. And we asked several physiological questions. One of the interests for me is under stress condition, how plants fix carbon. That's what drives yield. So we looked at the photosynthetic rates at 23 degrees under well-watered drought and 29 degrees heat and drought. And we can see a reduction in photosynthesis. Carbon fixation capacity reduces by anywhere at 29 degrees. There is a big drop, almost 40% drop. But I also looked at the carbon carboxylation rates here, like efficiency for Rubisco, VC max. Again, it drops. And the big drop is the rate of electron transport chain. And we also found like differences in conductances, whether it is conductance of CO2 into the substomatal cavity and conductance of dissolved CO2 all the way to the site of carboxylation. Here I'm showing the resource use efficiencies, particularly for carbon and nitrogen, again across all 4 stresses. On x-axis, I have stomatal conductance and integrated water use efficiency measured through carbon isotope ratios. And we can see as the stomatal conductance increase, the carbon isotope discrimination also increase. That means water use efficiency goes down in those ones. Under dry conditions, your water use efficiency improves. When it comes here, the x-axis is mesophyll conductance. That is how efficient is your photosynthetic machinery. And when we look at nitrogen isotopes and photosynthetic machinery, they were inversely related, as dry conditions don't let nitrogen to be picked up to above ground. and that leads to higher discrimination values that we observed. I was also interested what goes on during daytime and nighttime. Here I took two canola contrasting phenotypes that exhibited drought tolerance, and I subjected them to night and day cycles, and every 2 hours we measured how much water loss they do. Here there is a big difference between a drought sensitive and drought tolerant line. What I was very much equally interested is Curtis Rampel mentioned that we want to use canola in brown soils. What we need for brown soils is reduced nighttime transpirations, because as nighttime temperatures warm, we need to reduce that unproductive water loss in the absence of photosynthesis. This is one of the traits very much selected for Mediterranean climates when it comes to barley, wheat, and others, a trait again underexploited in canola breeding programs. And then we looked at some of the amino acids and hormones, again as stomatal conductance, what was driving the reductions or higher stomatal conductances. Here, this is cysteine is one of the sulfur-based amino acid. With increase in cysteine level, the stomatal conductance for water losses was coming down, and that followed a similar path like ABA hormone leading to stomatal closures. We were very happy. For last 2 days, I heard like oil content goes down during stress. Why oil content goes down during stress but protein increases? Here, your sulfur is the central molecule when it comes in Brassica for stress resilience. There are other molecules like prolines, GABA, and others, but here, the sulfur, instead of being always directed towards oil synthesis, when there is early drought stress or heat stress, it gets redirected to form a sulfur-based amino acid. That cysteine molecule moves to the guard cells and increases the ABA content of the guard cell, leading to stomatal aperture closure. With that stomatal closure where they conserve water, you reduce your photosynthetic fixation, because once you close stomata, you can't take CO2 in. That the only way that can sustain photosynthesis is by using its primary sugar as osmoprotectants of the cell, and that in turn will have negative effect on yield. So this was what was going on, the sulfur being redirected to deal with stress. If there is a biotechnological mechanism where we can avoid a lot of this path direction and move this way, then canola plants can produce much better oils even under stress condition. And we looked at few other metabolites. Here, in the interest of time, I will talk about only serine, which is a photorespiration. This is one of the molecule which is very unique. Serine is produced both in the cytosol and in the plastid. So the Brassica plants have figured out the very importance of serine, having to utilize both the pathways to sustain the photosynthetic machinery and the osmotic, cellular osmotic tolerances in the cytosol. We looked at waxes, and there is a big difference in wax morphology, but not in much differences in wax composition between tolerant and sensitive lines. In summary, I would like to mention that under warm climates, industrially relevant traits like taste, freshness, and shelf life may be affected due to high saturated levels of oils. Curtis Rampel mentioned that brown soils are very much the focus. So I thought I should just put like a couple of traits that as a physiologist I can bring for discussion as far as establishment is concerned. Canola hypocotyl length can be a trait for that to select for, and radical vigor. With that, I would like to thank the funding agency like Agriculture Development Fund, SAS Canola, and Agriculture Agri-Food Canada, and my colleagues Isabel, Steve, and Sally for providing the NAM population. With that, thank you.
Thank you, Raju, for a really good overview of the effect of stress on canola and an indication of how complex it all is. So our next speaker is going to be presenting to us as a pre-recorded presentation. And our speaker is Dr. Sarah Schiessl-Weidenweber, and she's a researcher and a lecturer at the Justus Liebig University in Gießen in Germany. Her background is physiology, biochemistry, and genetics. And she's going to be talking more about the role of genetics in abiotic stress, particularly drought stress, and transgenerational effects of stress. So if we're ready, we can launch our talk.
Welcome. Today I want to tell you a story about drought resistance in Brassica napus, not only in the first but also in the second generation. Why do I need to talk about drought? Climate change is increasing the probability of seasonal droughts also in Europe, and this will affect timing when drought is mostly devastating. So drought at flowering has much stronger effects than it has during the vegetative phase, as you can see here with the yield levels. Unfortunately, this is exactly when drought will happen in future. So the first peak of drought probability is in April, which is majorly the time where rapeseed fields are flowering in Europe. Unfortunately, there was quite little data when we started this project about drought tolerance at flowering. Most experiments were performed in early vegetative stages. So our questions were to identify factors which would contribute to drought tolerance in oilseed rape during the flowering time. And we also wanted to identify some possible breeding targets which could be used for improvements. And in the end, we also came to a side question in the beginning. We were asking ourselves what this drought stress would do to the seeds produced from the stress mama plants. I will talk first about the first question, about factors which contribute to drought tolerance in oilseed rape. I will therefore first explain how our experiment was set up. We did a semi-controlled container trial. The idea was to have a very large soil volume, which would allow to measure drought stress with a normal root development. The containers were basically garbage containers where we filled in some soil to mimic normal soil structure. The containers were placed in a wire cage, which was covered by a transparent roof to avoid precipitation, so we could control it. But the outside temperatures in wind and sunshine were as normal in an outside field. Soil capacity could be controlled using a specialized scale. So by weighing the containers, we could assume how much water is in there. The stress we performed was a 3 weeks trial. We just took those containers out of watering. So the stress increased steadily. So we had in the beginning a quite mild stress, but after the 3 weeks a quite severe stress. The stress treatment started shortly before flowering. The flowers had opened, so all plants would really have the stress during the flowering. The experiment was performed with 8 different genotypes, which were selected from a representative diversity set we had in-house, where we had some data about flowering time and drought tolerance in the early vegetative stage, and we selected genotypes which showed contrasting extremes there. We phenotype those plants for 36 different traits, among them life cycle traits, biomass, plant architecture, and yield components. And I could now show you a lot of bar plots about this, but I decided to summarize data in this kind of table. To explain it to you, what you see on the left panel is a short code for the trait. So you see beginning of flowering and end of flowering. Then you see plant height, dry mass, number of side branches, total number of siliques, grains per silique, 100 grain weight, and yield. As the most important traits to discuss now. For the plant architectural and life cycle traits. You see two arrows in this table. The first one always refers to early stress, the other one to late stress, apart from the flowering, which is beginning and end of flowering. Filled arrows show changes in this trait which were significant. Gray arrows show changes which showed a trend but were not significant. So I'll guide you through this table. So what did we see? We saw that all accessions have delayed end of flowering. So all of them increase the flowering duration. All accessions had a reduced plant height, quite strongly reduced and sometimes, as you have seen before. And most accessions, except two, had a reduced biomass already at early stress, but also at late stress. This points to a drought avoidance and water saver strategy, which is already described in the literature. For 5 accessions, we found a significant decrease in yield, while 3 accessions did not reduce yield significantly and were therefore regarded as tolerant. We grouped the accessions by their typical cluster of responses. We found 2 accessions which were both sensitive, which had smaller grains, so their yield loss was due to smaller grains. And we had 2 accessions which had smaller and less grains and less side branches. One of those genotypes could compensate the smaller and less grains with a higher number of siliques. So more siliques were formed. I will come to this in a minute. The next cluster did not show any changes about the yield components, so there were no changes, but one of the genotypes was sensitive due to a non-significant trend in the number of siliques. The last cluster had larger grains and did also not reduce biomass, so whilst the larger grains were obtained by no reduction in biomass. So we asked ourselves what were the success strategies now, so what turned those accessions into tolerant accessions. We found that for 2 of the tolerant accessions, they actually showed a later beginning of flowering than under control conditions, which points to a protective effect of a shift in flowering, which moved the pollination phase into a better watered environment and allowed more successful pollinations. The last tolerant accession had no reduction in grains per silique, so it could wisely nourish more grains per silique. This could also be linked to the no reduction in biomass, but wisely for the sensitive one, this was not enough. So to summarize, the success factors under drought were either an escape strategy, so escaping drought by a later flowering, or a better source-sink ratio, so a better distribution between biomass and seeds was obviously helpful to attain drought tolerance. However, this seems not to be predictive, so just later flowering is not the only factor to confer a drought tolerance, and also source-sink ratio alone was not helpful alone, so there must be another level of drought tolerance, and we assume this to be molecular. So we went on to check on possible breeding targets. What we did for that was to perform an RNA-seq. So we sampled the plants at early and at late stress, both over well-watered and the drought treatment, and then determined the gene expression level by mapping the reads back to the genome. And the gene expression levels were then clustered in a weighted gene co-expression network analysis. So to make it understandable to you, what did we do? we clustered the gene expression values by genotype. And if we have a group of genes which showed a similar expression profile, this is a gene module. Each of those gene modules was then given a color. And this color was then used to recognize it back on the plot. So what did we plot? We plotted the significance of correlation of this expression profile to a trait. So in this case, it was yield over the coefficient of correlation. So the more out to the left or far right, you can see that this is more significant and also more correlated. So the interesting modules are those on the far right or far left, because it doesn't matter if it's negative or positive. And we found that the processes which were linked to those gene expression modules were basically response to desiccation, RNA metabolic processes and gene expression, RNA methylation and modification, and photosynthetic processes and chlorophyll. Which makes a lot of sense because this is exactly what is expected for mechanisms to drought adaptation. And we were interested in which to which degree this would have been protective. So we compared the degree of up or down regulation in the intolerant and the sensitive accessions in the same cluster. So for example, here we compared this sensitive accession to tolerant accession, and we found that indeed the tolerant accession has a more strong upregulation of the gene expression of protective genes. In total, we found 600 genes which obviously had a protective effect. This is one of the best candidates. So this is a gene which is involved in the nitrogen starvation signaling pathways, obviously has a role in also regulating drought stress. Other genes which were more upregulated and tolerant than insensitive ones were involved in reactive oxygen species scavenging in the innate immune system and in chlorophyll synthesis. So to sum this up, the breeding targets we could identify were approximately 600 possible targets, which belong to aerosol scavenging, photosynthesis, the immune system, and nitrogen signaling pathways. So this would be interesting to follow up. In the last part of the presentation, I now want to talk about what we then studied on the next generation. So in the beginning, it was quite a side project, but resulted in a quite interesting publication then. So we basically wanted to know what would happen to the seeds which were grown on stressed mother plants. Would they show differences in germination or seedling development in the next generation? And what we did then, we were first checked how the quality of those seeds has changed. You might not be surprised, which is a known fact, that oil content in drought-stressed in seeds from drought-stressed mother plants goes down and the protein content goes up independently of the accession. All accessions have reduced oil content and increased protein content. The good news is that it didn't affect erucic acid content at all, and it had only very slight effects in certain accessions and also not consistent for the glucosinolate content. So the glucosinolate content was increased in one accession and decreased in another, but otherwise unchanged. the pattern of fatty acids also changed for some accessions. So 3 accessions reduced the content of oleic acid, 6 accessions had more linoleic acids, and 3 accessions more linolenic acids. So this obviously has an effect on the quality, but when we compare this to which accessions were indeed tolerant, so those with the gray arrows here in the yield, in the yield column here, then you see that there is no correlation in quality reduction and quantity reduction. So widely tolerant genotypes do not have a better seed quality. So this has to be addressed differently. So the next thing which really interested us then was that we saw that when we germinated those seeds and then measured the seedling vigor, we realized that plants which were stressed gave rise to seedlings which were bigger. So we had quite strong increases, about 30 to 40% in seedling vigor in the next generation. So here you see it in the bar plot that those plants in red, which arise from mother plants under stress, are markedly bigger, and this was also significant. We were, of course, interested if this has any correlation to yield, but again, no. Resistant cultivars were not more or less vigorous in the next generation. So unfortunately, drought tolerance obviously does not influence the vigor in the next generation. We also did not find any conclusive pattern in the germination rate. So we measured germination rate. So we found increases under stress in 4 genotypes, but these were completely different genotypes from those which were sensitive for tolerance, so the level did not play any role, and also mean germination type, both under control and under stress conditions, was completely uncorrelated to vigor. So neither is vigor correlated to yield, nor is it correlated to germination characteristics. What we did find was correlation with some metabolites. So best correlating was SMCSO, where the change rate in SMCSO and the change rate in fresh weight was quite well correlating. And we also found quite good correlations, for example, for glutamine and malate. So some metabolites are obviously well correlated, as you see here in the table. This can go back to the same cause. One of them might be the cause of the other, so we cannot fully explain if the metabolites are the cause or the consequence of the higher vigor. But it remains to be studied. So if you have any ideas for good cooperations, we would be very happy to find us in a project with you. So to summarize, we found that drought tolerance improves either by shifting flowering time or by improving the source-sink ratio. However, this alone is not predictive. Molecular protection mechanisms like aerosol scavenging and photosynthesis regulation seem to play a more important role in improving drought tolerance. The drought-stressed mother plants obviously give rise to seedlings with a higher vigor, but this is independent from the germination characteristics and is also independent from the level of drought tolerance of the generation before. With that, I want to thank a very, very big crowd of people who made it possible that this project and further projects were performed. And I thank you for your attention, and I'm happy to take questions.
Thank you, Sarah, particularly if you're online. So our final speaker in this session is Professor Keith Adams. So Keith Adams is a professor at the Department of Botany at the University of British Columbia in Vancouver. And his interests are genome structure, polyploidy, and alternative splicing. And he's going to talk about that in the context of abiotic stress in brassicas. So Keith, if you're ready. The floor is yours.
Hello, can you see and hear me?
Yes, yeah, we can see you and we can see your slides. Do you want to make them full screen for us?
Yeah. Okay.
Perfect, thank you.
Okay, everything is all right? Great.
Yep, looks
okay.
good.
First of all, I would like to thank Mark and the other organizers for inviting me to speak today. And today I'm going to be talking about a project that a former graduate student did in my lab. It's looking at transcriptomics of canola, particularly alternative splicing under heat, cold, and drought stresses. So first of all, I'd like to give a short introduction about polyploidy in plants. This is a topic that my lab has been studying for a number of years. Polyploidy is a very important and ongoing and recurrent process during plant evolution. As all of you know, canola is a nice example of a polyploid, but there are also a number of other cultivated plants that are polyploids. These include such plants like durum wheat and bread wheat, as well as others like cotton, peanut, coffee, potato, etc. So quite a number of important crop plants are polyploids. But in addition to that, most diploid plants have one or more polyploidy events in the evolutionary history of their lineage. And in the Brassica lineage, there are over 3 rounds of polyploidy that have occurred. One that is specific to the Brassicas after their divergence from the common ancestor with Arabidopsis. But then there are also 2 other ancient polyploidy events that have occurred in the common ancestor of Arabidopsis and Brassica. This one is thought to have taken place at the base of the Brassicaceae family, and then a slightly older one that encompasses a few other families within the Brassicales order. There are also a couple of even more ancient events, one that is shared by most eudicots and one that's shared by most angiosperms. So due to these multiple polyploidy events that have taken place in the Brassica lineage, it's one of the reasons why Brassicas have so many duplicated genes. This is the Brassica polyploidy triangle. A lot of you have probably seen this before. It's a diagram showing the 3 polyploid Brassica species that have been derived in different arrangements from 3 diploid Brassica species. And I have highlighted at the bottom Brassica napus, which is derived from a Brassica oleracea as well as Brassica rapa. There's been a lot of interest in studying molecular aspects of polyploidy in Brassica, as well as in a number of other polyploids, such as wheat and cotton and others. Some of the phenomena that people have seen in studies, in Brassica studies, as well as in other polyploid studies based on RNA-seq analysis, these include biased expression from one subgenome. So that is that for individual genes, the copy from one subgenome is expressed at higher levels than the copy from the other subgenome. Also, the phenomenon of subgenome dominance, where looking across a large number of genes, more of them have a expression bias towards one of the subgenomes or the other one. People have also seen that there is organ and tissue specific expression patterns, and also cell type specific expression patterns. And it's also been seen that expression levels are affected by abiotic stresses. Now I'd like to introduce the phenomenon of alternative splicing. Alternative splicing is an important aspect of gene expression that affects many genes in plants. This is when more than one type of final transcript is made from a single type of pre-mRNA by differential splicing. Some of the major types of alternative splicing events include intron retention, where an intron is retained in the transcripts, alternative 5' or 3' splice site usage, and also skipped exons. In plants, intron retention is the most common type of alternative splicing, and skipped exons is the least common type. So alternative splicing has a couple major roles. One is that it can create multiple protein isoforms from the same gene. The other major role is that it can regulate gene expression, in particular down-regulating gene expression by a phenomenon known as a nonsense-mediated RNA decay. This is when some of the transcripts, particularly ones with, but not exclusively ones that have premature stop codons, get degraded, and that lowers the overall level of expression or silences the gene. So now I'd like to talk about a project that a former PhD student in my lab, John Lee, did. He was looking at the effects of abiotic stress conditions on expression and alternative splicing in genes in canola. Some of the questions that he asked in the project were how do abiotic stresses affect expression levels of genes from each of the subgenomes? Also, how do abiotic stresses affect the alternative splicing patterns of genes from each subgenome? And do drought, heat, or cold stress have the greatest effect? And so these were the three stresses that we looked at in this project. So the way that this was done is that we used Brassica napus, the century summer rape cultivar. These were grown in growth chambers. At about 3 weeks, John did the stress treatments to the seedlings. These were done for 24 hours, the heat at 35 degrees, the cold at 4 degrees, and then the drought was done with 25% PEG to simulate drought conditions. He then collected the first true leaves from the plants and used 3 biological replicates for each genotype and stress condition. He did RNA extractions. The RNA was sequenced on Illumina HiSeq. This was done by Genome Quebec with 100 base pair paired in reads. And then John did a variety of data processing and analysis steps with a variety of different software packages. So I'd like to present a few of the results. This is a project that was published a couple of years ago in the journal Plant Genome. So of the stress treatments that we looked at, John found that the cold stress had the greatest effect on expression. It also seemed to have effects on different genes than the heat and the drought stresses did. You can see that on the left figure. This is a heat map looking at expression levels of about 1,000 different genes that had the strongest expression responses, and then grouped by the different subgenomes indicated by CT and AT, as well as the different stress conditions. And you can see that the overall heat map patterns for the cold stresses were somewhat different than for the heat and the drought stresses, indicating that the heat and drought had more similar effects on expression levels, and the cold stress was rather different. We also found that when grouping these together and doing comparisons of the different stresses and the different subgenomes, that indeed the cold stress had the different effects on gene expression, and also the largest effect compared to the cold and the drought stresses. And that's shown in this kind of, I guess you call that sort of a beige color right here that you see in the heat maps along here. We are also interested in the subgenome dynamics of the differentially expressed genes. John showed that more of the genes had a higher level of expression from the A subgenome than from the C subgenome of those that were differentially expressed. He also showed that there were more genes that were downregulated in response to the stress conditions than were upregulated, and that's shown in this figure on the right here. John was also interested in looking at the alternative splicing patterns in the genes. And this is a table that's showing the total number of genes that he looked at for alternative splicing patterns and whether there was an increase or decrease in levels of alternatively spliced transcripts in response to the stress conditions. And he found that the cold stress for both the AT subgenome and the CT subgenome had the largest changes in expression levels in response to the stress conditions. And also that more genes showed decreases in alternative splicing event levels than increases. This is a figure that's showing the overlap of the intron retention events in particular, showing intron retention events because this is the most common type of alternative splicing in plants and also the most common that we observed in the data set. It's showing the overlap of the drought, cold, and heat stress, both in terms of increased levels of alternatively spliced transcripts and decreased levels. You can see that a lot of the genes were specific for particular conditions, especially for cold stress. But there were some genes whose alternative splicing levels increased or decreased in two of the stresses, like here, or even a smaller number that were increased or decreased in all three of the different stress conditions. John also looked at the correlation between expression levels and alternative splicing events. And he found that for these four different alternative splicing event types, the intron retention, alternative acceptor, alternative donor, and alternative position, which is basically where both the 5' and the 3' splice sites are different in the alternatively spliced transcript, he found a negative correlation between expression and alternative splicing levels. Okay, so now let's say a little bit about the implications for these changes in alternative splicing event levels. It's been shown that in Arabidopsis, that levels of alternative splicing events in certain genes can have an effect on genotype. So it's possible that some of these changes we detected may have some phenotypic effects, which would be interesting to look at in the future. Also, some alternative splicing changes upon stress treatments may be helpful for the plants to cope with stress. In the future, it'd be interesting to do some functional studies with some of the genes that were identified in the study. We're also currently looking at effects of abiotic stress treatments on a resynthesized Brassica napus line, as well as its parents. And my lab has also been interested in the effects of biotic stresses on gene expression. We're completed in the process of publishing a project looking at sclerotinia infection and its effects on expression and alternative splicing. Hey, finally, I'd like to thank John, who was shown in the middle right here. He was also helped with some of the analyses by a former grad student, David Tack, and finally, NSERC for funding. So thank you for listening, and I'm happy to take questions.
Thank you very much, Keith. And thank you to all of our speakers. So that brings an end to the presentations, and now I'd like to open the floor now for questions and answers. So for those online, I will read the questions, or we'll read the questions to you. So do we have any questions from the audience? Actually, I think I'd like to start one straight away. Keith, you were talking about alternative splicing. Did you see anything different about alternative splicing in coding regions or non-coding regions? Because I know some genes have regulatory introns. Have you looked into that?
Yeah, that's an interesting question. We actually didn't look at the coding versus the non-coding regions, but that would be something interesting to look at to see if there was a difference there.
Yeah, thank you. Okay, so questions from the floor? Must be some very quiet crowd. Otherwise, we can shift to the online questions. Cathy, do you have anything?
We have.
I got one.
Oh, Keith. So Keith Downey has a question.
Oh, okay. This may be kind of silly, but has anybody looked at protoplasm? Protoplasm, you know, has effect in the cold reduction. I don't think we should completely ignore that component from this aspect of heat and cold response. So has anybody got an opinion on that?
Do we have any comments?
I'm not sure, Keith, regarding the protoplast, yeah, when it comes to cold. Yeah, maybe Keith can answer.
Any thoughts, Keith? I put you in the hot spot.
Yeah, my lab hasn't looked at protoplasts, but that could be interesting to do.
Do we have any questions online, Cathy?
We have one question for Raju. You mentioned a number of desired traits in canola varieties for the brown soil zone. Can you prioritize what a breeding program should focus on?
Well, I just presented 2 traits there, so I ranked them in that order of my preference. So hypocotyl length will be something like, because if we place the seed in much deeper profiles of soil, let's say 7 centimeters deep versus 10 centimeter deep to hit that moist zone, then having a longer hypocotyl length will allow the seed to come out, and by the time the roots will just reach the soil zone. So that's where, like, that has been successfully shown when it comes to coleoptile traits in wheat programs, where they put them at deeper soils in Australian conditions. Selecting for longer coleoptile is where their breeding programs are heading. And if we are serious in black soils, then hypocotyl length is what we should be focusing for.
What was the second trait they are asking?
That's related to the root vigor, radical vigor, how fast the radical goes and hits that soft spot of like accessing moisture. Yeah.
We have another exciting question on the chat. Can I ask that too?
Yes, please.
Okay. We heard this morning the canola hybrid marketplace may need more workhorse, like yield stable hybrids to support the racehorses that yield amazingly well under ideal conditions. Physiologically, what does a workhorse variety look like in a variable environment?
Well, if I can comment, Keith, you can join me here. For me, like selecting for heat tolerance is something we have to do when it comes to Brassica species, because if there is no flower set for seed formation, then whatever other traits we have will be of no much use. So the first and foremost is like, I look at like how arid countries like Australia are selecting for, they're not looking for drought tolerance. They're all selecting and breeding predominantly for heat tolerance, because once they get over heat, by the time they can always deal with drought one way or the other way.
Would you like to add anything to that, Keith?
No, I don't think I have anything to add.
I guess another question for everybody is whether we should be thinking more about teasing apart abiotic stress detection as well as the effects of stress on these plants. Does anybody have any comments on whether there is work in that area? Maybe some plants are more able to detect a stress at an earlier stage and respond appropriately. Any thoughts from the speakers?
Well, lot of like when I look at like NAM population that has been put together at AFC, we see some of the canola lines are very conservative, some are very much liberal. That means independent of whether there is stress or no water, heat, they continue to perform what they are doing and they get stressed out really fast. Whereas some, in spite of abundant water, they tend to ration their water use very, very judiciously. So we see the spectrum, but drought being such a dynamic trait, like, you know, the duration, the length, and what stage it comes, it's hard to just choose one or two that will decide drought. However, if I can add, like with my interest in carbon isotopes, the first wheat variety that was released at drought tolerant by physiologists used carbon isotope techniques. And the Drysdale is the wheat variety that was released in Australia. And the second variety that was released was from my breeding program with poplar, where we released drought tolerant poplar for prairie conditions. So integrating these long-term signals of carbon, oxygen, and nitrogen, one should be able to understand what goes on from emergence all the way to maturity. So more and more programs in Mediterranean environment are driving using these isotope signals.
Okay, we have another question in the chat.
Okay.
Do we know what variation exists for nighttime water loss among currently used canola varieties?
In general, if I talk crop systems between monocots and dicots, monocots have anywhere between up to 35% of water loss. Oh, when I say water loss, from emergence all the way to maturity, 35% of water is lost at night in the absence of sunlight. So that is unproductive water loss. If you save that water, say instead of opening your stomata that much, if you close fully, then you can save per day so many millimeters of water. When we multiply that millions of hectares that's planted, that's almost like extending your stress by a week, because you can save that much water, which would have lost unproductively. When it comes to canola species, I'm trying to characterize those, what is the range and how much it happens. And we need, like now Isabel and Steve Robinson has this lysimeter where you can leave the plant from emergence all the way to maturity on a balance. In my case, I was measuring every 2 hours, day and night. So someone worked during day, I worked at night, things like that. But now they have much more sophisticated, and we can tease apart which line loses water and whether they close partially, whether they close impartially, or whether they continue to lose water at night. So we should be able to separate out for the diversity that exists within our populations.
There's another one for Keith.
Okay, there's one more, Keith.
Is there a reason for using the cultivar Sentry that was registered in 1996? Does it have a unique quality or purpose in the lab?
Well, we actually used that cultivar because we had it available. There wasn't any anything special about that particular cultivar. One thing I think it would also be interesting to do in the future would be to look at a variety of different cultivars, because I have a feeling that the some of the stress responses will probably, at least responses to of gene expression will probably vary somewhat with different cultivars. So I think that would also be interesting to look at.
Seems like Sarah has joined us, and we have a couple of questions for her.
Sarah, welcome if you're here. We do have some questions for you, even
Thank
though it's
you.
9
Sorry
o'clock
for
at
being
night.
late. My baby just threw up.
Oh, great. Thank you. Good luck. Okay, so we have a question.
So for those higher vigor progeny lines, did they end up having a higher yield at the end compared to their lower vigor siblings with the same genetics? Does this mean that the source to sink relationship was compromised in that generation?
We actually did not go that far. The reason was that when we started the experiment, we did not plan to go to the next generation. So these were open pollinated seeds. So therefore, the genetics would not have been preserved. So we are planning to repeat the experiment to answer exactly that question.
Thank you. I think we do have one more for you.
No, I asked, both of them were the same, so I
Okay,
asked both
so
of them.
a pressing question, there was two the same. Do we have any more questions before we have probably like 2 minutes left? So if we have any more questions from the floor, now's your chance. Yes, so Katie has one more.
Okay. Yesterday, we heard about glowing plants and how excited the precision ag community is about it. How do you think this could be used in stress tolerance breeding programs?
You mean like the precision agriculture or the glowing plants?
I think the person is asking about the glowing plants.
So are they asking about plants that kind of call for help when they're under stress? Is that what they mean by glowing?
I wish they could explain
Well,
what
It'd be
they
very
mean.
interesting.
Yeah,
But we could have some
I
bright
can't
nights.
speak for glowing plants, but at least plants produce a lot of volatiles. When it comes to forestry, like they can pick like some of these gas samples and figure out whether the particular plant is stressed, and they have these volatile organic compounds that you can capture, and you can sense like whether a particular forested area is stressed due to a particular insect or stress, and one can understand those. So maybe if we are interested, like instead of going for glowing plants, maybe setting up these volatile compound traps in fields and look at what kind of volatiles the plants are producing to communicate that they are stressed. That's underutilized in agriculture systems when I compare that with forestry.
Okay, thank you. In the interest of time, I'd like to thank all the speakers for their really interesting presentations. This year, in lieu of speaker gifts, we're giving donations to the Keith Downey Undergraduate Scholarship Fund at the University of Saskatchewan. So thank you for your donations and thank you for your contributions. And I will draw this session to a close. You now have a quick coffee break and then we're back for the last section of the day. So thank you, Sarah and Keith, very much for joining us online. And Sarah, good luck with the little one.
Thank you so much. Have a good day.